All right, everybody. Thank you so much for being here. We will start today's webinar, the first one of this autumn semester. Thank you so much for everybody that's joining today to learn a little bit about how birds, bugs, and resistance kind of come together from the hand of Evangelos Mourkas, the department coming from the Department of Medical Sciences here at Uppsala University. Just a couple of things to mention before we start related to UAC. You know that the website has changed. So now there is no possibility to subscribe to a calendar for UAC, which means that the events that we do are not going to appear automatically in your calendars like they used to do. So you are going to have to add them manually somehow to your calendars when I send out emails about the events. I will try to find a way that maybe I can do an attachment and you only have to double click to add it to your calendar if you want to attend. But just have in mind that before they used to appear in your calendar automatically if you were subscribed, but that calendar has been finished with the change of website. So now there's a little bit more of steps to take in order to put our events on your calendar. As other webinars, we are going to run for about 50 minutes, the talk that Evangelos is going to give, and then we're going to have a Q&A session in the end. You are welcome to write your questions in the chat if you want me to give them to Evangelos in the end, or you could also wait and raise your hand during the Q&A and post them yourself. Just have in mind that we are recording this webinar in case you don't want to be on camera or on the microphone, then write your questions on the chat. And with that, it's my pleasure to introduce you to Evangelos Mourkas, that is going to talk to us a bit about his path, how he is studying the things that he's studying now, and how birds, bugs, and resistance come together and the impact of urbanization on pathogen spread. Thank you so much, Evangelos, for agreeing to share your work and your research with us, and the digital floor is yours.
Thank you so much for the invitation, Eva, and for the lovely introduction. So to begin with, let me share my screen. So as you probably noticed, I have joined from two computers. So maybe if everyone does that, then we'll have more people. Right. So can someone confirm that they can
Yes,
see my
I can see
screen?
your screen
Yes.
well, and you yourself as well. Yeah.
Perfect. So I will also switch my camera off so you can focus on the images in the screen rather than my face. Okay. So yes, my name is Evangelos Mourkas. I'm a researcher in infectious diseases at the Department of Medical Sciences and also a visiting researcher at INBIM here at Uppsala University. And as Eva mentioned, we're going to talk about birds, bugs, and resistance. So if you're interested in either of those things, or you just want to see some cool photos of birds, I guess you came to the right place. So for people who are interested in birds, this is not a seagull. Well, it's a northern fulmar, and the picture was captured on northern Iceland. Why is it not changing? Okay, now it is. So a little bit of background about myself. So I'm originally a vet. So I did my veterinary medicine degree in Greece back in 2012, where I graduated, before moving to Uppsala for the first time in 2013 to do my master's in infection biology. And this is really where I started getting into the world of research and start thinking about hypotheses and how we can answer hypothesis-driven questions in different research settings and in different projects. And then for some reason, I thought it would be a nice idea to do a PhD. So I moved to the UK to do that in the University of Bath, in the lovely city of Bath. And you can see that the model I've been holding in the two first images has changed during my PhD. So instead of furry or feathery friends, I've been mostly holding my laptop, really delving into microbial genomics, so looking at these tiny microbes and their genomes and trying to understand what's going on. And after that, I did my first postdoc in Bath in collaboration with the University of Leicester, working with Neisseria meningitidis in the UK. And I moved to Oxford for another postdoc at the INEOS, Oxford Institute for Antimicrobial Resistance and Research, before coming back to Uppsala for the second time as a researcher and working with Yusuf Yerhut and Patrick Elstrom at the hospital in Akademiska here, and also in the Zoonosis Science Center group at the group of Åke Lundqvist at INBIN. So a bit about my research and what are my interests in research. So I'm really into like pathogen genomics. So trying to see in the sequences of the different microbes together with traditional microbiology in the lab, try to see how different bacterial populations are behaving in the lab and what's in the actual genome that is causing them to do so. And then, of course, evolution. So all of these bacteria, they've been around us for many years and they have been evolving and they emerge and they cause diseases. And of course, the spread of antimicrobial resistance. So really understand the evolution of different bacteria, but also antimicrobial resistance with them. And much more recently, trying to look at the different populations of carriers versus disease using these genome-wide association studies that some of you might have heard. They've really been around and been used in humans. So you have a control population that's healthy, and then you have a cohort population that has disease, and then you're really comparing the genome to try to find hotspots that might explain the different phenotypes. So this has been implemented in the last 10 or so years in bacteria, really trying to answer the same thing. You have different populations, and for example, one is sensitive to antimicrobials and the other one is resistant. Can you really identify what is the underlying mechanism in the genome? So what's really causing it? So all of these years, my theme or my main interest in animals hasn't changed. It's just went from treating the actual animal into focusing on the bacteria that it carries, and especially these bacteria that they end up in the food products that we eat and cause disease in humans. More recently, being involved with a lot of projects at the hospital, looking at infections caused by bacteria following a surgery, for example, when you do a hip replacement. AMR in general, in all of these settings. So how does it spread? Where do you find it? Why is it there? Why is it a problem? A lot about pathogenicity and virulence, Neisseria meningitidis, or E. coli, Staphylococcus, all of these pathogens. Why can they cause disease in humans, and under which circumstances they are able to do so? So these are all the different publications I've been involved in. Some of them I was leading all of these years, and I have highlighted all the different countries next to its paper. So this is really just to highlight that research is about talking with other people, collaborating with other people all over the world. So all of these years working in research, I have made a lot of connections and friends and collaborate with so many different people and expertise all around the globe. And this is really how research should be, and it should be shared with everyone. And talking about the world, how many of you have actually thought of how does the world that we live in look like when it's nighttime? So you can get a lot of images from Google, but it's really like looking at the lights during night, and then you take a picture from a satellite and you can actually visualize that. So the modern world is changing, and this is at an unprecedented pace. It's going so fast. So in just the last 100 years, the cities that we live in, they have expanded at a really rapid rate. And you can think of all the urbanization that's going on, the skyscrapers, highways, how the landscapes that wildlife is living is being transformed and it's not really looking the same. So as cities expand and natural habitats are shrinking, this is forcing wildlife to live closer and closer to humans. But why do we care about wildlife in urban environments? I mean, you picture yourself in a very busy city, you're walking around, all people are rushing by, you have lots of buildings rising above. And amid this jungle of concrete, you might notice a pigeon pecking some crumbles at the Craigs, at the Oxford train station. Or if you take a walk in Hyde Park, you'll see lots of different birds there eating food and just mingling with humans and themselves. And talking about wildlife, you might notice a fox urinating in a sewer just down the street where you, at the time you return home from your work. So all these things are happening around us, and these birds, these animals are more than just city dwellers. They are a part of a delicate balance that sustains our ecosystems. And whether they are living in a city or in a forest, these animals, they tell us something about the world that we live in. And why focusing on wild birds? So what's so specific about these animals? These animals, they have been resilient survivors for many, many years, traveling hundreds, thousands of kilometers. And you can see in the map, these are some of the migratory routes that the wild birds take, traveling all around the world, changing countries, continents, and hemispheres. And they've been doing it so successfully for so many years. But how many years exactly? So how old are wild birds? So birds are actually modern day relatives of small carnivorous dinosaurs. And scientists have discovered that birds evolved from dinosaurs thanks to fossils that they found in places like China and South America. And there are some really famous fossils that show that feathers, like birds, like here, but also features like teeth and the long tail, really showing the transition between the dinosaurs and the birds. So over millions of years, these early birds, they have evolved features for flying, like feathers, lighter bones, and wings that they could create thrust. So the feathers that were originally used for warmth or display, they later became a key for flight. And eventually these ancient birds, they gave rise to the diverse bird species that we see today. And really, now with the advanced technology, we can use genomics also, sequence all the genomes of the birds and try to calibrate when different bird species diversified back in the day. So this is just a take-home message to think that wild birds, they're really, really old in evolutionary terms. And there are so many different species, like just illustrating the diversity of these animals in nature now. And just a small, not forgetting about bacteria here, they're going to come up hot very soon. But in terms of how old things are in the world that we live in, prokaryotes, which is the bacteria, are really, 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 really old. So these are around here, and then you have the birds here, and we are somewhere here. So as we talked about, wild birds aren't just, they're very important for the roles that they play in the ecosystems. They're pollinating plants, they're controlling pests, they're dispersing seeds, but they're also powerful indicators of the health of the planet that we live in. So as they move across borders, ecosystems, they come in contact with different diverse environments, and then they're carrying with them vital information about those ecosystems. And as we said, they don't really have passports. They don't stay within national boundaries. Their movement is tied to global phenomena like trade or animal migration or weather patterns and even human displacement and conflicts. So talking about the One Health, where we have the environment, humans, and the animals, in a rapidly urbanizing or globalized world where travel and trade connect us more than ever, birds are exposed to various environmental stressors and also various pathogens. And this reflects the broader global health trends. So, for instance, birds might encounter antibiotic resistant bacteria in agricultural areas or pollutants in industrial zones or even novel pathogens. So really by studying the bacteria and the viruses and all this stuff that these birds carry, we can gain some insights into how global issues like climate change and conflict and human activities are shaping the health of the ecosystem. And we have a lot of different stressors. Yes, WHO has put all these lovely things together just to remind us. Urbanization, we have deforestation, biodiversity loss, climate change, agricultural pollution, migration, and everything. All of these are interconnected challenges that are fundamentally reshaping the ecosystems that we live in. And as our cities continue to expand, we are witnessing more and more interactions between wildlife, humans, and livestock. And of course, this opens the door for the spread of zoonotic pathogens. So pathogens that are transmitted from humans to animals, from animals to humans, sorry. Yes, so we're now going to focus on intensive farming and just have a few graphs just guiding our narrative and really showing some data that it's just really surprised to see and not many people realize. So we're just going to take the US as an example here, and we can see here the food per person in kilograms that they eat in the US, and how has this changed over the last 100 years or so. So we can see a rapid increase in production of chicken. And this is up to 2010. So I assume this is just continued going up until now, this is 2024. And of course, this means that we are having much more chickens around because the demand is high and we don't have enough chickens. So we need to produce more. And this has also increased, and there are some predictions following this line for 2050. And this is millions of tons of production on this axis. So yes, we need more chicken, and we need it fast. So we need to grow really big chickens in such a short period of time. And companies have been selecting traits in the breeds that can actually do so. But what does that mean if you think of a chicken, an older chicken, and how the chicken looks like today? This is how a chicken looked like in 1957. It was a bit less than a kilo. And you can see the transition over the years from a bit less than a kilo to 4.2 kilos now. And I don't even want to think how it's going to look today in 2024. And that comes with a consequence. And the immediate consequence is that chickens, they outweigh all other birds now. So if you add up the mass of all the birds on the planet, 71% of this weight belongs to poultry. And if you think about it if you put this into context, it's insane. And this brings us to the next problem, which is the loss of biodiversity. This is a nice graph put together by a friend and colleague, Dr. Merrick, showing the number of of birds on Earth. So wild birds, I mean, of course, we can't have a direct estimate, but we have an approximate guess estimate based on observations. So there are around 200 to 400 billion wild birds in the world, and there are 15 to 20,000 species of wild birds in these numbers here. And then we have the chicken. So the chicken, based on the FAO stat estimates, the population of the chicken has increased. And in 2020, it was around 30 billion chickens in the world. And we're talking about one species here. So basically, if you put that into numbers, roughly it's 1 to 10, 1 to 9 birds in the world right now is a chicken. And they don't roam around, flying around like the wild birds. They all live in farms. They're kept together in close quarters. And really, these large-scale agricultural systems designed to feed growing human populations, they have dramatically reduced the biodiversity that we see. So instead of the diverse habitats, we see fields dominated by single crop or huge livestock operations. But intensive farming doesn't just affect biodiversity, it also promotes the spread of pathogens. But I forgot to mention that we have one species here, but actually the two breeds that are supplying all the different farms in the world, they come from these two different breeds. So Ross 308 and Cobb 500. And one is in the UK, the other one is in the States. So in farms where livestock are kept in close quarters, yes, the use of antibiotics is common, or used to be more common. And this, of course, leads to the emergence of antimicrobial resistant bacteria, or AMR. And these resistant bacteria don't just stay confined to farms, they can spread to wildlife, including wild birds. So again, taking as an example the US, I don't know why US has been the scapegoat here, but I promise it's not on purpose, maybe. But the production or the use of antibiotics for pigs in 2011 was 4 times more than the ones used in humans. And then the FDA decided to do something. So in 2016, they put a ban, introduced a ban for the use of antibiotics in healthy herds and flocks as growth promoters. And then that's the numbers afterwards. So we see there is a reduction, but we're still not there yet. There's still much more to be done. And we have the urban environment, we have frequent use of antimicrobials in healthcare, in agriculture, we have the waste management challenges, and all this leads to more antimicrobial resistance. And more of these resistance bacteria are found in urban environments. And there you have urban adapted birds like pigeons or crows, gulls, that are in constant contact with these environments. And this makes these perfect carriers for these types of bacteria. So these birds interact with human waste, they scavenge in landfills, and they come in contact with contaminated water sources. And as a result, they may spread these resistance bacteria across large areas. And just to not point the finger at the US, looking at the sewage or environmental contaminated water sources, that's another indicator of how AMR how is the picture of AMR worldwide. So there are regulations in the US and in Europe and in some countries about the use of antibiotics, but in a lot of other countries, and especially in low middle income countries, you can see like Brazil, a lot of countries in Africa and the Southeast, Middle East and Southeast Asia, you have a lot of different antimicrobial resistance in the sewage. So this kind of reflects the situations in the world, and it's quite scary that you have so many different antibiotic resistance in the sewage worldwide. And of course, a lot of people have seen something similar to this. Is we have different antibiotics to treat infections, but then you develop or invent an antibiotic, and then after a while you have resistance, and that's happening over the last years for all the different antibiotics that we have in the market. So what do we see in these photos here? We see a clear impact on the gut microbiomes of these animals. So they are roaming around in the landfills, and you can see the pelicans or the starlings here. They're adapting to this new environment, and this is helping also the bird species that have scavenging behavior to further develop this. So this constant exposure, it affects the diversity and the composition of the bird's microbiomes. And this often leads to the presence of more pathogens and more antimicrobial resistance that we shed and ends up in the landfill and in contaminated sources. So, for example, birds like starlings or gulls, which they thrive in urban environments, are known to carry a high number of AMR genes in the microbiomes compared to rural species that are not found there. And this just shows us that urbanization is just affecting where birds live, but it also affects the bacteria that they carry, with potential serious consequences for public health. So this is one of recent studies that we did, and we actually tried to put all this context together and really try and see, can we use a bacterium as an indicator in different wild birds to see how their microbiome is being affected, and do they really carry resistant, antimicrobial resistant enteric pathogens. So this in the middle is our model. It's called Campylobacter jejuni, and I'm going to talk in a bit of what it is and what it does for people that they don't know. So it's important to keep in mind that birds are old. In evolutionary terms, much older than chickens and livestock animals. And we also talked about the origin of bacteria, which are even older. And why do I say this? So Campylobacter jejuni is one of the most common causes of foodborne associated diarrhea in humans. And we humans, we do not carry this in our gut. We eat most of the time contaminated food sources, like chicken, that we don't cook well, or we wash in the sink, and then we get cross-contamination in our utensils and things like that. But the point I want to make in this photo is that this bacterium, it naturally exists in the microbiome of different animals and in different wild birds. So we see if you take all these genomes of these different bacteria from the different animals and you make a phylogenetic tree and try to see how closely related different genomes are, you end up with these clusters of genomes or isolates, which are commonly associated with a single host. So you can see here with orange, all these clones are associated with chicken. And then you have some blue, which are associated with ruminants. And then you have some other ones, black, that are associated with wild birds. But the interesting thing about wild birds is location doesn't matter. So if you're a blackbird living in Sweden or a blackbird living in Australia, and you carry Campylobacter jejuni in your gut, these will cluster together, even if you live thousands of kilometers apart. So there's a really strong host signal with C. jejuni and the host, and particularly in wild birds, because we think they have been evolved or co-evolved for billions of years together. And of course, there are other pathogenic bacteria that have been and can be found in different bird species. Some of you might have heard of Salmonella, Acinetobacter, or E. coli. By the way, this is amazing what AI can do. You just tell it, make this figure for me and this picture for me, and it will do. It needs some more work with the spelling, but it's getting there. But Campylobacter is a very good model to use if you want to see how healthy the microbiome of the bird is, because it's naturally found in most wild birds that we see in nature. So what we did in the study, we got access to a lot of different isolates and genomes from 30 different wild bird species. And then we grouped each bird species by families or orders where they belong. And then we made the phylogeny to look how closely related these isolates will be from the different wild birds. And we see on the tree that there are some clusters associated with a single host, like here with gulls, or here with dunlins, and some in crows. But there you have other parts of the tree where you have clusters from multiple bird species. But what does this actually mean? So what we call this coevolution of particular bacterial clones or bacteria in C. jejuni with particular bird species, they have evolved this relationship where they live together and really adapted to live in the gut of a particular wild bird. So in theory, you should only have one bird species with its associated clones, which we call the specialist. But for some birds, what we do see is that they carry genomes from that particular clone, but they also carry genomes from other clones. And these clones are quite interesting because some of them are frequently associated with human diarrhea. And if you remember the previous picture, some of them are found in livestock animals. So why do we see this in some birds? Is it something special about the bird that leads to this behavior? So what we did is we focused on some ecological host traits from different bird species. So how big is the bird? What's the number of eggs that it lays? So the clutch size. The mating system, is it a monogamous bird? Is it a polygamous bird? Does it migrate locally? Does it not migrate at all? Does it go through continents? Does it like water, or does it stay in the forest? What's the diet? Very important. But what does it eat? And what's the social organization? So all of these different traits of the birds. And then we try to have, using our Campylobacter jejuni clones, try to quantify some microbial traits. So how many different lineages do you see in a bird species? How many different AMR markers do you see in a bird species? And what's the actual proportion of resistant isolates that you see? And then we try to do associations to see, do we have any correlation with any of these traits and any of these traits for a particular bird species? But there is something very important missing. How close birds live to humans? And of course, that can be very objective. You go out of your house now, what's the most probable, that's the bird that you're most likely to see outside? Are you going to see a dunlin, which is a shorebird, or are you going to see a crow or a gull? But that is very subjective. We wanted to put a number behind how close humans live to birds and vice versa. So we came up with a method using human and bird abundance data, and I'm not going to go into detail how this method works, but briefly, for a particular latitude and longitude point, you have an abundance score for humans and for birds, and then you try to overlay this using formulas, published formulas, and then come up with a metric that shows how close birds live to humans. And again, I like the US. This is an example of two birds living in the US. And in the maps here, you can see the black dots representing where humans live. The red dots here representing where this beautiful bird lives. And the yellow dots representing where the crows live. So you can straight away see the overlap between where humans live and crows live in all different areas compared to the mountain bluebird. So we did our correlations, and the strongest correlation association that we found was that the closer you live to urbanized areas, closer to humans, the more lineages you get, different lineages, and the more different AMR genes you get. And this has been summarized in a follow-up article pointing to our paper, which shows all of these potential transmission routes that you have. So you have birds here that they really like water, birds that are scavengers, you have humans. So, and the idea being these birds in the microbiomes, they have lineages they shouldn't have. And they have them because they get exposed to them, because these lineages are associated with human diarrhea, and humans get them from livestock. And a lot of these lineages, they are antimicrobial resistance. So this really shows like a metric of lineage diversity and AMR gene diversity in particular bird species correlates with how close these live with humans. So solutions. You have a lot of different images here. We can talk about reducing the antibiotic use in agriculture, surveillance of AMR worldwide, including the wild birds into the picture, developing new drugs to tackle antimicrobial resistance. It's a global coalition, funding, of course, rapid diagnostics and global public awareness, very important. But this really isn't the one person's or one discipline's problem to solve. This is a much larger scale, and we really need people from different disciplines working together, from clinicians, veterinarians, biologists, microbiologists, but also people involved with sewage treatment, city planners. You know, all of these different people, they need to come together and come up with effective strategies to tackle some of these problems. So I did promise some cool figures and pictures. So I'm going to talk about a small project that we have run in the Arctic. And again, this is a northern farmer. So we really wanted to look at the antimicrobial resistant enteric pathogens in Arctic birds. But why in the Arctic? These are the three different locations where we took samples from. One of them is in Iceland, at the northeast part of Iceland. The other one was in Finland, but really where Finland, Norway, and Sweden meet. And the other one was in Norway. So the idea is that in these really remote, pristine habitats, there shouldn't really be any antimicrobial resistance or any of these non-wild bird lineages. And I did visit 2 out of 3 of these sites myself. So just to put some perspective of where the actual station is, and it's a small village called Raufarhorn. Well, it probably called something different in Icelandic, but I was never able to pronounce that. This is the main road of Iceland for people who have been there, and you need to go that way to reach that. And there isn't really much around there. But just a bit of context. At one point in time, this small village was home to the largest export harbor in Iceland. So around the 40s or the 50s, where the herring frenzy dominated the Icelandic economy, Ralph was an important place in that economic chain. I think the locals told me that there were around 3,000 or 5,000 people living there. But after the herring were fished out, the effect was devastating for the village. So I think it had around 150 inhabitants when I went, but even less of that during winter. So it's really not very common for someone that to do everything from going sampling and looking for the different animals, capturing them, taking the fecal sample, culturing the sample, and doing all the microbiology and genomic work after that. I just want to point out, being in the field myself, how difficult it is to get hold of these type of samples. So this is me and Oscar exploring the terrain, looking at the different animals. You can see this is a common snipe, abundant in Iceland during the summer. And of course, you end up looking at some other animals that I haven't seen before. So this is an Arctic fox. But as I said, it really takes a lot of time to explore the terrain and the behavior from different birds. And if you're not there with people that are experts in the field, like Oscar and Jose, who is taking the photo but is not in the picture, you can't really do without them. So as I said, this was a small fishing village, and there's still fishing activities going on there. And you'd assume this is a very pristine habitat with no urbanization or human activities messing with the ecosystem. But let me switch to no laser point so I can play this video. But have a look at this. This is happening at the port of the village. Oops. There we go. And I gave a hint already of what that may be. So yes, these are fish guts. And that's, if I remember correctly, one of the 20 crates that were where people did that specific day. So yeah, this is not normal, and this is not supposed to happen. And if this happens in those remote areas, imagine what happens in very urbanized areas. So you have a lot of different wild birds coming together to eat these fish guts. And then again, really pointing out at how difficult it is to take these type of samples. You have to track the birds, understand their behavior. You can take some feces from the ground, but really in Iceland, there's nothing. It's just tundra. So you can try nets, you can try traps, but these birds, they're not stupid. They will just fly around and avoid the net and the traps. So so many different bird species there. It's amazing to first come in contact with an Arctic tern and understand how protective they are of the colony and their nesting grounds. So they will attack you if you go near, and they will aim for your eyes first, and it's really scary. This is a red phalarope, another very exciting bird. And of course, we had all the ethical permissions obtained to do all this type of work and sampling there. So the way we did it was mainly using remote traps. So you have to track the different birds and see where they nest. And then when appropriate, you can set a remote trap in the nest, and you can trap both individuals there. And you place them in this little bag, in that little how do you call it, net little thing, and then the bird usually poops within a minute, and then you can do all sorts of measurements, ring the bird, and release it back. And then we take the feces and we do we freeze it and store it in particular solutions to further work with it in the lab. So here you see ring plover, Sutherland, and I don't remember the name of this bird. But I do know that these birds, they travel thousands of kilometers to come to Iceland. It's fascinating creatures. And yeah, this is me holding the Sutherland. So a few things about the other side in Kilpisjärvi, and as I said, it's really close to the 3-point CERN between Norway, Finland, and Sweden. And here we're talking about different types of birds. You have all these small little forest birds, blue throat, very really like a hallmark of the region. People were very happy and proud to capture this. So I was there with a group of people from University of Helsinki doing a field course for 2 weeks. So they were regularly capturing the birds using nets in the forest and ringing them, and I was tagging along, helping and collecting samples. So yeah, blue throat, this is a common red poll, a red wing, and some sparrows. And then you get the samples, you go back in the lab, and you culture this case culture for Campylobacter, but we also save a specimen for metagenomics to really see what is there. And some of these samples are sequenced, so there's work ongoing to analyze if they have any AMR and what lineage they are. Are they supposed to be there or not? So really excited about the results. So to conclude, as we're reaching our time, what can we learn from birds? So we talked about urbanization, intensive farming, the rise of AMR, and all these interconnected challenges that are fundamentally reshaping the ecosystem that we live in. And as the cities continue to expand, we're having these frequent interactions between wildlife and humans and livestock, as we talked about. So yes, birds are, specifically those living in urban areas are adapted there. They are at the forefront of this shift, of this reshaping of the ecosystem. So they carry not only the microbes of their own environments, but also those of the human world. And really to mitigate the risk of zoonotic diseases and the spread of AMR, we really need to understand how these systems are linked and how we can better manage the impact of human activities on the natural world. So yes, interconnected ecosystems as one health and understanding the dynamics. A lot of different people involved, and I don't know where to begin with thanking different people, but particularly Jose, who has been instrumental on the bird side of view and discussions and really leading this project together with me. And then, of course, Sam and Ben at Oxford, and a lot of other people in various universities. And the previous group in Oxford, but also the current group in the Zoonosis Science Center. And of course, thank you all of you who joined us today and had a look at this talk. And I'm really happy to take any questions. I can already see there are some in the chat.
Thank you so much. Thank you, Evangelos. It was a great talk. I think you could put your camera up now. Yes, great. So we can
Yes.
see you. Yes, so Elizabeth says thank you so much for a very nice presentation. We'll leave some time for you guys to maybe write some questions in the chat, but also if you want to ask Evangelos directly a question, you can put your hand up. So I will give you the voice to give to ask the questions. But because people will probably are thinking about it now, I will ask you a question to start with and then see if we can get the discussion started. I was curious to know, once you in your first project, you were looking at see what things kind of correlate with the data you found, and then nothing really could really make sense of what you were getting. And then you thought about setting up this urbanization kind of index or how far away things are from the birds from people. What challenges did you guys face when trying to set up such a metric or such a way of measuring this?
Oh, that's a very good question, and it really hits a nail there. Um, it was actually we started thinking about the different metrics of the birds, and we did see some weak correlations between diet, for example, or mating system, and some of these microbial metrics. And originally, we thought about having a subjective metric of uh, high urbanized or very close to humans, medium, and then not close to humans. And we ran the analysis using this, but that's very objective. And we really wanted to come down with a more accurate metric. And it actually, the idea came when we were in Iceland. We were trapped in the station for a week because there was a snowstorm at the beginning of May, and we couldn't really go outside. So we started looking at we know there are abundance of human data where humans live in particular geographical locations. And then we thought about eBird. So eBird is a website where a lot of bird watchers, they go in and they observe a bird and they give an observation. I saw that bird in that spot, and they have to put longitude and latitude information. And then we actually checked the data for the different bird species that we have. It's amazing how meticulous and accurate these data are. It's amazing. We compare them with the different with the literature, with migration patterns. Jose has done a lot of work on this. Make sure this is as close to what we
Reality.
see naturally as possible. Which is we were very happy with with that aspect. So yes,
So
thank you
you,
for asking this. It's a
so
lot.
you
It was a lot of work. Yes.
You could consider this in part a bit of like citizen science kind of project, right? Which is because all that data is actually collected by bird watchers and people that are not really part of the research team per se, but is actually utilizing a resource that is out there done by, hobbyists in a sense.
Exactly. I mean, we did have to request the data from eBird, but they had no problem. They just gave us all the data. We're talking about millions of observations for each bird species.
Did you think of expanding this? So, for example, now that you have a metric and you have, you know, we researchers like to think about numbers also helping us to predict things. So, is this new way of measuring urbanization and birds' closeness to urbanization can be used for prospective, like kind of looking into the future and predicting what might happen? Have you thought about that?
We have thought about the application of the tool in research in general. Maybe not specifically this, but that's a brilliant suggestion. Um, what we initially thought is we will develop this method, and then everybody that wants to answer any questions using that, they can do. Because the way this works is you need latitude and longitude coordinates and the score of abundance or score of whatever observations, anything. And that can be, in this context, for example, what are the coordinates of different farms? And then you have the birds. So you can measure how different bird species live to farms, for example.
Mhm, mhm.
Or it can be, I don't know, anything, plants and birds, plants and humans, anything where you have the data, you can use the method. And we're really happy to put that online.
That's great. That's a very new thing that could potentially, people can brainstorm about how it can be used for the particular projects if there's anything that they might be able to use. So Cecilia is writing, thank you. Are these birds in the north not migrating? And what about natural resistant to antibiotics since almost all antibiotics are appearing as natural products? I was also
Mm.
thinking a little bit about that, like on your second project, a little bit about how are you going to know what does that resistance, if you find it, means? Does it mean a selection? It means that it's been there forever? If you have thought about that, yeah.
That's a brilliant question. Depending on the migrating or not migrating, it depends on the species. And to be honest, I'm not the most qualified person to talk about this. That will be Jose. I believe most of the birds are migrating, and we did factor that into the analysis using transcontinental local migrants and non-migrants. So that has been described, but exactly like species and it's not clear in my head because that's Jose's area of expertise. But what I can say about the natural resistance to antibiotics, we know that Campylobacter is naturally resistant to beta-lactams. It carries a lot of these beta-lactam genes, and this is why we excluded them from the analysis. So the idea being that there is resistance to other antibiotics, tetracycline, and this usually comes with a cost. So carrying a gene that gives you a resistance to an antibiotic, it usually comes with a cost. And we really have these gene markers that they shouldn't be there. And we really see them in livestock animals and in the hospitals. So if we see any of these markers present in our isolates, then that's scary. And if we don't see any of these in birds that are not close to humans, then that's a strong indication that, they might come in contact, but because they are not frequently exposed to these, they don't really set in to stay in the bird. So one might ask how long a bird keeps the resistance whilst it's been exposed, but nobody has done that. This is really like you need to do a very controlled. I haven't read about it. I know some people are trying to do it in the lab in vivo, but in controlled populations, and you have to recapture the same bird individuals. It's complicated. I hope
Yes.
that answers.
It's pretty cool. Yeah, I think so. It's looking at things that, you are sure that if they're there, they might have come from a separate contact, and then try to see longitudinally how long this might stay there. Um, Pretty cool. So Isis is just writing a remark. Thank you for an insightful presentation. A good reminder that tackling AMR requires a One Health perspective. The environmental health and our ecosystems are paramount as well, which, yeah, I think your presentation really brings it together. And then Walla is asking, I'm wondering if it's possible to detect the development and transfer of AMR through birds during migration through following data from different protected areas around different countries.
Let me read the question again. I mean, yes, that is possible to do, but then again, it comes down to how difficult it is to get hold of this type of data. So you will have to get to the protected areas and then capture birds, and then make sure you capture the same population of birds uh, during the migration pattern. So you have to really have a lot of funding and a lot of people with different expertise doing this. And Jose is actually doing a similar project in Chile in a smaller population where he's recapturing the same population, sometimes even the same birds along the migration, because Jose is from Chile, and Chile is like very long. It's a coastal country, really goes from like Patagonia in the south all the way up to Peru and Atacama in the north. So he's been doing this. So hopefully in the future we can answer some of these questions or have like an insight of what's going on.
Is the migration patterns also a bit similar in Sweden, since it's also a rather long country like this? Do you know?
It depends on the bird, I think, but I would assume there are long migration patterns, and
In Sweden.
birds that end up here, they come from Africa
Yeah, that for
and
sure.
other places. Yes.
That's very cool. Thank you, everybody, so much for your questions. If there is no more questions in the chat or with hands up, then first, I will thank you again, Evangelos, for bringing this to us. If you liked the talk and you want to learn a bit more about Evangelos' path and insights, I'm going to interview him for our podcast. So in the near future, there will be an interview as well for you to listen to.